I'd really like to convey today is what you can do with DNA barcoding and how you can do it practically at home and why it's so cool. And I'd like everyone to understand how to do it and how to do the data analysis. So if you have any questions about anything I say, put them in the chat. And after the talk, I'll go through the chat. And I don't have any time constraints here. It's like 10 p.m. Here in California. I usually stay up pretty late, so I am happy to sit here and answer all of your questions and talk about DNA barcoding, and until the last question is answered and until every last person here understands everything that I know about it. Just put them in the chat and I'll go through the chat once I'm done talking. And so I also have my friend Mandy here sitting next to me, and she loves mushrooms. She goes mushroom hunting every single day. She lives up here in Humboldt County, which is where I am now. But she has not done any DNA barcoding. And in fact, I sent her her first DNA sequence from something that she found just last night. So she's very new to it. So I told her that if there's anything that I say that isn't easy to understand, just to interrupt me. So if you hear another voice in here, that's Mandy asking about this. Let's see. So here's some resources. Get my slide advancer. Here's some good resources for sequencing stuff. The first one is a wiki page that I wrote. And it's a kind of a short wiki page, but what I put in there is everything you need to do the actual DNA barcoding. So there is a list of equipment at the end, and there's also the primers, the procedure, the exact chemicals. Really, if you just read this page, you should be able to do this all at home without my help. But I'm also really happy to help people get this working and analyze the data and just get the chemistry working, get the PCR working. So if you guys want to set up a lab, just send me a message and I can help you select equipment. I can help you find good used equipment that's likely to work, and help you get the PCR reactions working. I help you with the electrophoresis and the data analysis. I get a lot of messages, but most of the messages I get are like, what is this mushroom? Is it psychedelic? And I don't mind answering questions like that, but I'd really much rather prefer to answer questions like, how do I analyze my sequence, or how do I make my gel electrophoresis work? So don't hesitate to send me messages, emails, and I will walk you through this and just keep helping you guys out until you have as many working DNA labs as you like. Also, if you want my slides for this talk, that's the second link here. It's just this, I think it's a PDF. Yep. And then there's a Facebook group that I started called Fungal Sequencing. And it's a pretty cool Facebook group now. A lot of the top fungal sequencing people, even some really good professional mycologists are in this group. And so if you have any questions, you can post them in there, just general discussion. I also did one called Fungal Sequencing Results, and that's just for posting stuff you find. So if you get some sequences back, but you don't want to write a scientific paper, or even if you do, but you just want to really quick throw it on Facebook just to share what you've learned or share your data, you can post in Fungal Sequencing Results. And that is just a group that's specifically for that. And then I went through this whole DNA barcoding process and took a photo of each step of the process. And so it's about 40 or 50 photos, and I put them in a Facebook post. So that's the bottom link you see here. If you go to Fungal Sequencing, you don't have to scroll down very far to find it, but that just gives you an actual picture of every last step in the process. So to make DNA sequences, the first thing you do is extract the DNA, and then you run PCR, which stands for polymerase chain reaction. And then you use gel electrophoresis to check if the PCR worked. And if the PCR did work, then you do Sanger sequencing. Once you finish with Sanger sequencing, you have this text file, about 600 characters maybe, and you can paste that into NCBI BLAST, and that's when you start to get your results. Because a sequence means nothing on its own. It only is meaningful when you start comparing it to other sequences. So NCBI BLAST compares your sequence to everything else in GenBank. And then once you've done that, you can download all the closely related sequences and calculate a phylogenetic tree. And that's the most accurate way to visualize what it is that you've learned. So you can do this PCR DNA barcoding process pretty much anywhere. This is a home lab. It's in a kitchen. This is Counterculture Labs, which is a biohacker space in Oakland. And there's more and more of these biohacker spaces being set up. For a long time, there was traditional hacker spaces where people would hack computers and solder electronics. But in the past few years, biohacker spaces have been starting to get popular. So it's the same concept, except instead of hacking computers, you hack biological things like do DNA sequencing or genetic engineering or play with chemicals. And so this place is one of the largest biohacker spaces, and it's open to the public. So everybody pays a small fee to get 24/7 access to this lab. And then you can do whatever kind of science you want there. Or just work on your own project, or they have a bunch of projects that are kind of like community lab projects. They're trying to reprogram bacteria to make insulin and stuff like that. So most large cities have these biohacker spaces, and almost any biohacker space will have everything you need to do DNA barcoding, because PCR machines are kind of central to a lot of different things in biohacking. So I've visited biohacker spaces in Austin and Chicago and New York and Seattle. Most large cities have them. And if you can just join one of these groups, then they usually have all the machines and pipettes and all the stuff you need to get going. But you can also just order the stuff. It's not too expensive, and then you can just set up a lab at home. And that's what I did during the pandemic when they closed all the labs. This is Damon Teague's bathtub. He was doing PCR in his bathtub. I asked him why his bathtub. He said it was the cleanest place in his house. And then I don't know if you remember, a couple of years ago, the government of the United States shut down. So Damon Teague was going to teach a biohacking class, or DNA barcoding class, but the building shut down because it didn't get funding. So they just did the class in a local coffee shop. So you can set this stuff up anywhere. It doesn't really take very much space. Here is my car. And so I just put this piece of wood between the passenger seat and the gear shift. And then I have a table, and I can do PCR and microscopy in my car. And then I run the PCR machine in the trunk. So you can see over on the left there, there's a 2000 watt inverter to power the PCR machine. And when I do it this way, then I can just FedEx my PCR products into the Sanger lab and have my DNA sequencing results before I even get home. So I set these up at fungus fairs and all over the place. But of course, it's easiest just to set them up at your house. So they have a bunch of these little PCR machines that have come in the market recently. They're usually pretty small, so they can do usually between 8 and 16 reactions at a time. So that means you would be barcoding between 8 and 16 mushrooms per run. Usually I recommend people get a used PCR machine from eBay because for less money than these things cost, you can get a 96 well machine, so you can do up to 96 of them and get a professional thing. So the first step in this whole process, you just need to get your DNA into solution. So that way you're basically just grinding up the mushroom and dissolving it in there's usually some salts and some preservatives. And so I usually use these really cheap, simple DNA extractions that only take a few minutes. If you go into a university lab, you often find their DNA extractions take 24 hours for all the steps. But it turns out about 90% of the time, you don't have to do all that. And these quick, simple DNA extraction methods work just fine. And I can send you the recipes for this stuff. I even put it in this presentation. But occasionally there are chemicals in mushrooms, or more often in plants, that inhibit PCR, and they cause the PCR not to work. So usually if you run into a situation like that, you can just dilute your extract, and there'll still be enough DNA for the PCR to work, but there won't be enough inhibitor. But if that doesn't work, then you can do get one of these more expensive DNA extraction kits, and they will purify your DNA and remove anything that's stopping your PCR from working. So this is what I use 99% of the time. It's called Extractinamp, which is a commercial product, but this has been reverse engineered from the commercial product. And so it's very cheap and easy to make, and works really well. On fresh material, it works more than 90% of the time. On dried material, it depends on more things, but a good run is like 75, 80% of the success rate. And then if your samples have been sitting around for 3, 5, 10 years, you might have more like 30% success rate. So after a few years, the DNA breaks down. Though sometimes you can put a 100-year-old mushroom through this process and get a perfect DNA sequence. So there's two solutions: extraction solution and dilution solution. The extraction solution has tris, potassium chloride, EDTA, water, and sodium hydroxide. So it's basically just some salts and preservatives. And this you can store at room temperature. The dilution solution is a protein called bovine serum albumin. And this can get moldy or bacterial. So I store this in the freezer once I make this solution, and then just like store it in the fridge for like a week, and usually try to keep it sterile when I make it. Though if you're freezing it, you don't really need to keep it super sterile. And here's the procedure that I use. Basically, I take a little bit of mushroom, put it in a little tube, put in a little bit of extraction solution, grind it up, and then heat the tube for about 10 minutes at 95 degrees Celsius. I find if I skip that heating step, I get about 20% less success rate. And then I add equal volume of the dilution solution, and then it's ready to go into the PCR. So I use these forceps to grab the mushrooms, because one of the main things you want to avoid is cross-contamination. So if you get little bits of 2 different mushrooms in the same tube and run PCR on it, if they both work, then your data will come out complete garbage, because you have 2 sequences on top of each other. So I sterilize these forceps with fire, and then I let them cool on this rack below. And then I take a bunch of little tubes. These are 1.5 milliliter centrifuge tubes. You can also use like the 200 microliter PCR tubes. And then I number them. And it's really important to keep track of these numbers, because later when your sequences come back, you want to know for sure which mushroom it came from. So I'd break off a little bit of the mushroom and put them into the tubes. Now, the exact amount of mushroom you put in is not very important. You can put in like a really tiny bit that you can barely see. You can put in a whole lot. The more mushroom you put in, the more of that extraction solution you have to use. The only constraint is that you need to have some liquid in the tube when you're done with this process to put it into the PCR. So once I have a little bit of mushroom in the tube, then I grind the mushroom with one of these plastic pestles. And then I put the plastic pestles in bleach to clean the DNA off so I can reuse them. I keep really good track of which mushroom goes into which tube. You can see on this Ganoderma down here, I actually wrote the tube number on the Ganoderma. So usually I just write on the bag. So what I write on every single bag is the date, and then X, and then the tube number. And that way, if I get a sequence that comes back and I'm not sure that something maybe got switched, I can find the bag and be like, okay, this mushroom went into tube number 2. And that's really helpful later on. And also just going through my collections, that way I know which ones that I've processed. And if the PCR doesn't work, then I'll write again another date and another tube number. And if it's a really difficult sample, maybe the DNA has been degraded by bacteria or something, then there'll be like 5 or 6 numbers written on the bag. And at that point, I'm like, oh man, I got to give up. This thing is just not going to sequence. So here's the bleach solution. It's 10% bleach, which is really good at ripping apart the DNA molecule. And here's Conoculture Labs. And so I have a whole bunch of dried specimens, and they go into a whole bunch of tubes. Here's Jason Hollinger. He studies lichens, and he works. He's one of the admins for mushroomobserver.org. And he just took this pestle and put it into a cordless drill because lichens are really tough, and that way he can grind it up. Sometimes I do that for polypores, though usually you don't need to do this. It works just fine without doing this, even for lichens and polypores. So once you have your DNA extract, you run PCR. So this stands for polymerase chain reaction. And it was invented 30, 40 years ago by Kary Mullis. And what PCR does is it amplifies a certain part of the genome. So mushrooms have usually about 40 million nucleotides or 40 million bases. So that means that the DNA code of a mushroom is like a book with 40 million letters. But Sanger sequencing can do up to about 1,000 bases or 1,000 letters at a time. So what PCR does is it helps, lets you decide which 1,000 characters out of the 40 million total characters in the whole genome that you want to sequence. And so here's a PCR machine. And all a PCR machine does is it heats up and cools down the sample. And so when you're setting up a PCR reaction, you put in a forward primer, a reverse primer, the mushroom extract, and the PCR master mix. And the primers are pieces of custom DNA that are about 20 bases long. And what they do is they search through the whole genome for a sequence that's complementary to the primer sequence, and then they attach there. And then the PCR master mix has an enzyme that copies DNA, and it starts copying the DNA at the point where the primer attached. So these primers, they cost only about $5 to make. And by make, I mean you go to a web page and you paste in the DNA sequence of the primer you want, and they synthesize this piece of DNA with whatever you typed into that web page. And then this primer goes and searches through the whole genome for whatever matches. So you can make primers for anything. You can make primers for a certain gene that's really interesting to you, or you can make primers that will identify if the mushroom is in a certain genus. But the most common primer that you'll almost always want to use, especially for a first pass, is called the ITS. And I'll talk a little bit more about that later. I got some slides about that. But even the ITS primers, usually you don't buy them. Usually you just go to one of these web pages, Google for the ITS primer sequence, paste it into the web page, and then they synthesize the primer for you. Here's some primers down in front here. So they come in a tube like this. And when you buy primers, usually you get just a few micrograms of primer, and that is enough for about 2,000 PCR reactions. So the primer is really cheap. The thing that costs a little bit more in PCR is the PCR master mix. And the PCR master mix has all the chemicals to make PCR work, which includes the DNTPs, which is the blank DNA. So it's just free nucleotides that are floating around in solution. And then it has the polymerase enzyme, which does the actual work of grabbing the nucleotide out of solution and incorporating it into the DNA chain. And then some preservatives and additives that help make sure it works. So here's the recipe I use for all my PCR. The PCR is not very difficult. It's easier than baking bread, but more difficult than cooking eggs. So I put in like half of it will be water. So a total would be 25 microliters, which is about half of a drop of liquid. And so half of it's water. And then half of it's like the master mix, and then just a little bit of the forward primer. And the forward primer tells it where to start copying the DNA. And a little bit of the reverse primer, and that tells it where to stop copying the DNA. and then just a little bit of the mushroom extract. So 1 microliter is like a 25th of a drop. So you see, you're only using like a couple micrograms of the mushroom in your PCR. So the amount of material that it takes to do DNA sequencing is incredibly tiny. So all the PCR machine does is it raises and lowers the temperature of that aluminum block. And so first it raises it to whatever temperature you program it to, so you can put whatever kind of program you want in there, but you pretty much always want to use the same program. And that does 95 degrees Celsius, so almost boiling, and that melts the DNA. And when you melt DNA, the double helix becomes single strands, so it's free floating. And then that goes down to 55 Celsius, and the DNA sticks back together. But when it sticks back together, these primers will attach to the part of the DNA that's complementary to the primer sequence. And then the PCR machine goes up to 72 degrees, and that's when the actual copying happens. So the enzyme was found from this bacteria that was found in the really hot geothermal tide pools at Yellowstone National Park. And so this bacteria can survive in very hot water that would kill most organisms. And to do that, it needs a polymerase so it can do its DNA copying at that really high temperature. So the high temperature polymerase was the key to making this whole thing work. So each cycle takes about a minute, and then you repeat the cycle about 30 times, and each cycle doubles the amount of DNA that's in solution. So after you've doubled it 30 times, you take just a few molecules of DNA and you have billions of identical copies. And when you have billions of identical copies, then you can run it through Sanger sequencing and get your sequence. I used to run PCR on ice, so this is like a cooler. Turns out that it's actually fine for a few minutes at room temperature, but what I'll do is I'll set up 8 PCR reactions and then just throw it in the freezer until I'm ready to start the machine. I also used to do 96 PCR reactions at a time. That is cool because you're sequencing 96 different mushrooms, but it's a lot of pipetting and it's very repetitive. So these days I usually do more like 24, because that way I'm still getting 24 sequences back, but it's not nearly so repetitive and I'm not spending hours on the same step. So it's a lot easier. So once your PCR finishes, then you can use gel electrophoresis to see if your PCR worked. Now, if you have a lot of money, you can skip this step because you can just send it off for Sanger sequencing and not worry about whether your PCR worked. But Sanger sequencing costs between $3 and $8 US. So it's a little bit expensive, especially if you have a lot of samples. So gel electrophoresis can tell you yes or no answer, whether or not your PCR worked. And that way, if it didn't work, then I can rerun the PCR, maybe do another DNA extract or change something about the reaction to try to make my PCR work. And then if it does, then I don't have to spend my $8 on the reactions that didn't work. So what I do is I take the PCR product and take just a little bit of it, usually about a third of it, so about 8 microliters. I put it on a piece of parafilm. You can use plastic or metal, tin foil, whatever, it doesn't matter. And this is to mix the loading dye with the PCR. So loading dye helps the DNA be visible, and it helps it sink down into the bottom of the well. You'll see why that's important in a second. So here we are pouring a gel, and the gel is agarose, so it's a very purified seaweed, very similar to what you use for mushroom cultivation, but more purified. And so I make it in the microwave and pour it into this box. This is the electrophoresis gel box that you see here. And then I pour a buffer over the gel and load the samples into the buffer. So when I cast the gel, I have these combs that have 8 different tines. And so that makes 8 wells. And that way I can put 8 samples into the gel here. And so that is 8 different mushrooms. It's checking to see if the PCR worked. I usually have 3 rows of 8, so it's 24 samples, so it'll fit into the gel. And then I put high voltage DC through the gel for about 20 minutes. Usually about 100 volts works well. 200 volts also works and gives you your results about twice as fast. So what the high voltage does is this DNA is negatively charged, which means that it moves towards the positive electrode. So by using electricity, I can pull the DNA through the gel. And the gel is like a 3D matrix. And the longer pieces of DNA go through the gel slower than the shorter pieces of DNA, because the longer pieces are smacking into the matrix more often than the shorter pieces. So what we're seeing here is the result, and this is after I have thrown the gel onto a black light. These black lights are super dangerous. Don't ask me how I know. So people don't usually use them anymore, and now we use blue light. So the new gel stains use, instead of ultraviolet, just like about a 400 nanometer blue light, you can just buy a 490 nanometer LED. And that way you don't have to have this super dangerous black light that will give you cancer and sunburn and fry your eyes. But I took this picture a couple of years ago. So what I would do is throw it on the black light and then hold my cell phone over it, take a picture real quick, and then get out of there before it can give me skin cancer. And that worked pretty well. But in any case, these results show you is how well each PCR reaction works. So this is PCR reaction number 1, and there's a very thin band here. So this band is shows that there's a very small amount of DNA. And so if I try to sequence number 1, it may or may not work. I don't know if if it's something that I'm really interested in, it's like kind of maybe worth $8 to find out if it works. But if it's the less interesting mushroom, then I would just run the PCR reaction again and see if I can get a better band. And then this down here is the primer. The primer traveled farther down the gel than my PCR product. And so this is always there, and you ignore this bottom part. Now here is sample 2, and this has a really sharp band here. And what that sharp band means is that what came out of my PCR reaction was a whole lot of DNA that was all exactly the same length. And since I was pulling it through, pulling the DNA through the gel with electricity and separating it by size, if I get a lot of DNA that's all the same length, then it shows up as a band on this gel. And that means my PCR reaction worked. And if I send it off for sequencing, I'll probably get good data. Here's tube 3. There's absolutely nothing where you would expect to see a band. So that means that if I send the tube 3 off for sequencing, I'll be wasting $8. Number 4 worked really good. Number 5 looks good enough. I'll probably get a good sequence out of that. 6 and 7 worked really well. And tube number 8, nothing. I'll need to rerun the PCR on that one. So you can see it's a pretty good PCR run here. I only had 4 or 5 of them that didn't work well. And so that is a good day when you have that many that work.
Could you see anything without the light, like to the naked eye?
So Mandy asked if you see anything to the naked eye without the light, and the answer is no, because DNA isn't visible. So when I make the gel, I put in a DNA stain, and the DNA stain binds to the DNA, and it causes the DNA to be fluorescent. So this particular DNA stain is ethidium bromide, which is super dangerous and causes cancer, according to some people, although it never gave me cancer. But it requires ultraviolet light, and it's a hazardous material. So now there's a little bit safer DNA stains that are not quite as carcinogenic as ethidium bromide. And instead of being excited by ultraviolet light, they're also excited by blue light. So you can use nice, safe blue light, and it's not quite as hazardous. But no matter what kind of DNA stain you use, the DNA is invisible to the naked eye until you shine the light of the correct wavelength on it. And then it fluoresces, and you get to visualize the DNA on the gel.
Alan, just a question from Fran. When you repeat PCR, do you use the same amounts of reagents?
So the amount of reagent that you use is not terribly critical. I usually use 25 microliters. And when I send it off for sequencing, the sequencing places want 10 microliters. So that gives me 18 microliters left over to run the gel. And that if I mess up when I'm running the gel, then I can run it again and still have enough for sequencing. If you want to have a little more buffer, so you can mess up with the gel a few times, then you can do a bigger PCR reaction, like 30 or 50 microliters. Or if you're very confident in your pipetting abilities, you can go down to 15. So you have just barely enough to run one gel. But I pretty much always do 25 because it makes the math easy, and then I get to run 2 gels, and that works fine for me. So once you have a PCR product that works, the next step is to sequence it. And there's a few different ways to do this. You could buy a Sanger sequencer and then set that up and run it at home. The problem with that is that it's expensive, and usually Sanger sequencers come with maintenance contracts. So you sign up for like this person that comes out and services it. And also the equipment or the chemicals, the reagents you need to run Sanger sequencing, the big dye and all of that is pretty expensive, especially on a small scale. So it's a lot cheaper and easier just to outsource the Sanger sequencing part to a lab who does this full time, because they have big robots that just Sanger sequence all night, and they buy their equipment, they buy their chemicals at very large volumes, so they don't have to pay as much for the chemicals, and then you don't have to worry about your machine breaking or even buying a machine in the first place. So pretty much everybody that's not doing thousands of sequences per month should you use one of these services. One of the most popular ones is Macrogen in South Korea, and they'll probably charge about $5, and you just FedEx your PCR products over there, and about a day later you get your data in the email. In the United States, I usually use one called GeneWiz, which is nice and local, but they're a little expensive at $8 per reaction. Eurofins has the best deal that I know of. You can buy these 96 well plates for $350, and then you fill up the 96 wells and FedEx it in, and you get 96 sequences for $350 US. There's probably for you guys, you'll be wanting to send it to Macrogen because they're not super far from you, and they got good prices, and they've been doing this for a long time, and so you get good, reliable results from them. So why would you want to do all this stuff? Well, the DNA barcoding is really cool for a lot of reasons. One thing is that you can see how this thing that you found fits together in the whole tree of life. And it's really cool because the next time you find something and you have sequenced it, you have this increased insight that you normally can't get about stuff that you found, about what its closest relatives really are. And then you see something that you find in nature and you're like, oh yeah, I know that how that fits in. It's, you know, maybe it's right next to some European thing, or it's right next to some species that doesn't look anything like it, but actually is really closely related to it. So it's really cool, kind of like how microscopy gives you a whole nother layer of beauty in the mushrooms. The DNA barcoding gives you a whole nother layer of understanding on the mushrooms you found. But also just knowing where your blast matches are. The first thing you do when you get a sequence back is you run it through NCBI BLAST, and that compares your DNA sequence with everything else in GenBank. And the first thing you notice are the 100% matches, if there are any. So if you see some sequences that match 100% or very near 100%, then you know that somebody else has sequenced the same organism, and you can click on those sequences and see where in the world they collected it. And so that's really good information to know where in the world this same thing occurs, especially if your goal is taxonomy. So say you're trying to identify a mushroom and you get a bunch of DNA sequence matches from Europe, then you know that you should be looking at European names because it's very likely that somebody in Europe has described it. Whereas if all of your blast matches are in Australia, maybe a few in New Zealand, then you know that those European names are very unlikely to apply, and you should be looking for Australian names. Or if you don't get any matches at all, you know you have something that's pretty rare and obscure, and very likely local, because if it was something that was found in the United States or Europe, somebody probably would have sequenced it and put it in the GenBank, or maybe you found something rare and obscure. Either way, really cool find and really cool insights that you really can't get any other way. When I first started with mycology, I was wanting to discover new species. And so I bought a microscope in 2007, and I'm like, awesome, I'm going to use this microscope to discover new species. And so I started measuring spores. And what I discovered is that every time I found a mushroom and I thought I knew what it was, I put it under the microscope and the microscopy confirmed that it was what I thought it was. And after going through 50 or 100 samples, I'm like, man, this is no fun. All the microscope does is says yes, like it is what I thought it was. It turns out that I was wrong. It very often wasn't what I thought it was. The issue is that with microscopy, closely related species will have very similar or overlapping microscopic features. So microscopy is really good when you have two different things that are not closely related. They're very distant related, but due to convergent evolution, they look almost the same. The microscope, you can separate things like that in seconds. But when you have a closely related species in Europe and then you're finding in Australia, when you use the microscope, you're going to be like, oh yeah, I think this is what's in Europe. Or you'll be like, man, the spores here are just half a micrometer smaller than they are in Europe and North America. And then you're left wondering, how significant is that? Does that mean it's a different species because the spores are a little bit smaller? So the DNA sequencing gives you a lot more data and helps you answer these questions a whole lot more definitively. Also, microscopy is just really hard because you're looking at thousands of shapes and trying to compare them with thousands of other shapes that are in thousands of different scientific papers and books. And the interpretation of that is really subjective and really takes a lot of practice. Both doing the comparison and just preparing the slides and everything. Whereas the DNA sequencing, either you get your data or you don't. And so you get much more definitive answers. But the microscopy is still super useful. What I do is I almost never do microscopy on something until I get a DNA sequence back. And once I have my DNA sequences, all of a sudden, all the microscopy I do is way more meaningful. And it's more meaningful for a few reasons. One, if you're trying to separate different species with a microscope, then you're trying to find characteristics that are a little bit different, but every collection is going to be a little bit different. So you never know where to draw the line. And maybe after you scope dozens of collections, you start to get an idea of where to draw the line. But if you can sequence a bunch of collections that you found and then put them into two piles, one pile is one species, one pile is the other species, then you can use the microscope. And once you know for sure which is one species and which is the next, it's a much better use of time to try to figure out which ones are different. So once you get the DNA sequence back, it's not just a magic thing that tells you what it is automatically, though it is sort of close to that sometimes. So there's analysis that goes into it. And how you do the analysis is real important. So I'll try to talk a little bit about how to get the correct information out of your sequence. But a DNA sequence is just a string of letters, just looks like a paragraph. If you want all 40 million bases, that costs about $1,500. Though someone messaged me yesterday, told me he could get it done for $150, which sounds too good to be true. It probably is true though. But in any case, that would give you 40 million base pairs, which would be very unmanageable. And without anything to compare with, what do you even do with 40 million base pairs? So these snippets of DNA that are like 600 to 1,000 characters long are really useful because they are so easy to use and store and compare. So you get, in your DNA sequence, you have 4 possible letters in the alphabet, A, T, G, and C. And so each species has a unique DNA barcode. Usually, about 1% of the time, you'll have different species that have the same DNA barcode. So that's really good that you're able to separate species 99% of the time. Or rarely, the same species will have different DNA barcodes, but usually it works really well. And then here is an entry in GenBank. So once you get a DNA sequence, the really cool thing to do is to upload it to GenBank. And the reason why this is so cool is because the next time somebody sequences the same mushroom, they will see your match come up. And they'll be like, oh, wow, the QMS sequenced this same thing. So I know it occurs in Queensland as well. And then they can compare their sequence to yours. When somebody else builds a phylogenetic tree, they can use your sequences in their tree. So trees are always way more useful when there's more nodes, more branches in the tree. And these DNA sequences, they're going to be in GenBank forever. So in 1,000 years, none of us will be here, but all these DNA sequences will be in there, and people will be like, well, what was in Queensland 1,000 years ago? And they could look at photos, but photos are kind of ambiguous. So they'll be looking at these DNA sequences and being like, okay, now we know exactly how the environment has changed over the years, because we know for sure what was there. So this is a Rusula that I found in Michoacan, Mexico, and uploaded the DNA sequence to GenBank. And so when I get a sequence back, the first thing I do with it after I blast it and clean it up is to upload it to GenBank, because if I don't, I will forget, and then I have a huge pile of DNA sequences to upload to GenBank, and a lot of really valuable data that people have gotten is not available and not in GenBank because people just forgot to upload it. So anyone can upload DNA sequences to GenBank. It doesn't take very long. It takes maybe 4 minutes to upload a DNA sequence to GenBank, so it's totally worth doing. It also connects me to other people really well. So other professional mycologists, they'll sequence something, they'll see my blast matches, and then they'll message me and be like, hey, can you send me this collection, or can you do some microscopy, or you can find collaborators or other people working on the same thing. A really cool thing about this GenBank entry is I put a mushroom observer number on it. So 99% of what's in GenBank does not have a mushroom observer or iNaturalist number, and that means you don't really know what they sequenced. You can't get a photo of it. You can guess at what they sequenced based on who they are and how well they would know that genus, or just what they called it, but you don't really know. But what you can do is add your photos to Mushroom Observer or iNaturalist. And then if you like microscopes, you can add the microscopy to Mushroom Observer to iNaturalist. You can put that in one of these fields here, like specimen voucher. And that way, anytime someone blasts something and sees your entry, they will know that your entry is one of the entries that have photos, which will make your entry way more valuable than the entries of all of the professional mycologists, because the professional mycologists don't use Mushroom Observer and iNaturalist, so you can never tell what it is that they sequenced.
Is there a cost to use this?
No, GenBank is totally free. It's run by the United States government, the National Institute of Health. You can just go onto the GenBank submission portal and submit sequences. If anyone wants to know how to do it, I can do one live if we feel like it. But it's real easy. You just go there, fill out a few fields, and upload your DNA, and then it takes about a week to get into the database, and then it's there forever. I like to fill out as many of these other fields as I can. So organism is what I think it is. I could be wrong, but when someone uploads something as Rusula lutea, you can be pretty sure that it's a yellow Rusula. So that's good information. And then I put in the URL of the Mushroom Observer observation, so you can just copy and paste this, go and see my photos and all the other metadata, the microscopy, if I did it. And then I put in a country field. That's one of the most important fields, because I not only put in the country, but also kind of exactly where I found it. So in this one, it's the state of Michoacan, and I found it at the Reserva de la Biosfera de la Mariposa Monarca, which is the place that all those monarch butterflies go in the winter. So a really special place. And then I give actual GPS coordinates. So if somebody thinks that this is a really cool species, maybe it's a new species, they know exactly where to go to recollect it. Like put the altitude, my name, and when I found it. So here is a DNA barcode. And that DNA barcode I showed you was from the ITS gene. ITS stands for internal transcribed spacer. And the really cool thing about ITS is that it does absolutely nothing. And because it does absolutely nothing, it's one of the most variable genes. It is the most variable gene that people often sequence. So that means that when you have two species that are pretty closely related and you want to be able to tell them apart, ITS is the best gene to sequence because it's going to have the most differences. You'll have the most differences between different species, the best species level resolution. And it's also decided that the ITS is the international standard barcode for DNA sequencing, which means that pretty much everybody who sequences a mushroom, they'll sequence the ITS first, and they will put that into GenBank. So if you're trying to identify a mushroom and your question is, what is this mushroom? Or where else in the world does this mushroom occur? ITS is the gene that you want to sequence because that has the most entries in GenBank, so the most sequences to compare against, and it's the most variable. But if your question, I'll talk about that in a second. Here is a map of the ITS. So the 3' end is basically the left, it's where it starts. And then the 5' end is over on the right, that's where it ends. So these here are different primers. So if I put ITS1F into my PCR reaction, that means that it starts copying the DNA right here. And right here is the end of the 18S gene, and 18S is very conserved. And that's important because I need my primers to attach to a place that's very conserved, because if I try to attach a primer to a variable place, then it won't attach to most fungi. So almost all fungi have the exact same 18S sequence. So that gives the ITS primers a really good place to attach. If I want a little bit less data, I can put ITS5 in, or even a little bit less, ITS1, and then it starts copying a little bit later. Doesn't really matter because this is so conserved. But after this really conserved gene, we have a really variable gene. It's called the ITS1. It's about 200 bases or 200 characters. And so these next 200 characters are super variable. So if you have different species and you compare these 200 characters, you'll almost always see differences there. After that, you have this really conserved region. And the really conserved region is called the 5.8S. And that's about 160 characters. And after 5.8S ends, you have another really variable region called ITS2. And then after that, you have a very conserved region called the LSU, or large subunit. And so the LSU conserved region is really good because you could have your reverse primer attach in the LSU, and you have a stable place to tell it to where to start copying, a stable place to tell it where to stop copying. And then you get all this variable information in the middle, and that's what's actually taxonomically significant. So if you run your ITS, so usually when I run an ITS PCR, I put ITS1F as my forward primer. The F stands for fungi, which means that this primer ignores DNA that's not fungal. So it ignores human, bacterial, plant DNA. So that's really cool. And then I'll put ITS4 in for a reverse, and that will amplify this whole thing. Now, if that doesn't work, maybe because my mushroom was moldy or because it's 100 years old, I can put in a primer that binds in this 5.8S region and get just half the gene. And when DNA degrades, it breaks that chain of the double helix. And so it breaks it into shorter pieces. So if you try to sequence a shorter part of the genome, it'll be more likely to work, especially if your sample is degraded. So I can put a primer like ITS2 in, which amplifies just these 200 bases, and you're much more likely to get a sequence out of something 30 years old using the ITS primer, but you get a little bit less data back. So what you can do is you can use the ITS3 primer to do a forward read starting in the 5.8S to ITS4 over here, and get these in 2 separate regions. That way you're spending $16 instead of $8, but you're able to sequence really old or degraded DNA. So there's also other genes that people use, and the best gene to use depends on what your question is. Say you really like boletes, and your question is, how are all these boletes related? How do they fit together? ITS is a terrible gene to use because boletes are extremely diverse and ITS is extremely variable. So if you try to build an ITS tree, phylogenetic tree, using all the different bolete sequences from all the different bolete genera, the computer will get very confused, and it'll express that confusion with giving your tree very low bootstrap support. And what that means is that the computer is telling you, I have no idea how all these things fit together. I just, that your sequences don't line up because they're too variable. So if you're trying to do something like make a phylogeny of the boletes, or really a phylogeny of any large genus, then you want to use more conserved genes. So more conserved genes, they evolve much slower than ITS. So if you sequence 2 closely related species, but you use a more conserved gene, you'll get identical sequences back. So these more conserved genes are not a good gene to choose if your question is, what mushroom did I find? But to get a good phylogeny, you definitely want to use these more conserved genes. So the most popular one for boletes and a lot of other stuff is the LSU, large subunit. It's very close to the ITS. It's also very easy to amplify, so you'll have very good PCR success with it. And here's what the LSU looks like. Usually when I do LSU PCRs, I run LORR with the LR0R primer. And then I use the LR7 as a reverse. That'll usually give me like 1200 bases or something. If that doesn't work, I'll use LR3R starting right here with LR7. And that gives me less data, but less data is way better than no data. And then you have protein coding genes. These genes actually do something. They code for proteins. That makes them more conserved because mutations in protein coding genes often make it so the mushroom is broken and cannot reproduce. But occasionally, you can have a mutation in a gene that actually does something that's not fatal to the mushroom because it doesn't affect how the mushroom codes. Sometimes it even makes the mushroom better. So these genes are more conserved, and they're also from a very different part of the genome than ITS. So you're kind of getting a second opinion from a very different part of the genome. So the most popular protein coding genes are TEF1, which stands for translation elongation factor 1, RPB2, RPB1, beta tubulin. Though, really there are thousands, even hundreds of thousands of these genes, and you can sequence any one of them for the same, $8. But you might as well sequence the same ones that everyone else did, because that way you can compare your data to everyone else. You won't have to sequence each collection to make every node in your tree. So you pretty much always want to run ITS. And then if ITS doesn't answer your question, then run these other genes. 95% of the time, my question is, what mushroom is this? And how is it related to the closely related species? So usually ITS does answer my question. And when I sequence more genes, I'm just kind of disappointed because I have these cool sequences, but they didn't really tell me much that I didn't know. But if I was building a big phylogeny, then that wouldn't be the case, and these other genes would be super important. Here's some tools that I use to do all this work. These tools are actually pretty easy to use. There's just like some weird terminology that you have to figure out, but really you're just figuring out how things evolve and really what could be more simple than evolution. So after you learn the terminology and kind of figure out how to use these tools, they're really simple to use. You don't have to be like a PhD student or anything. Like I never went to college at all, and I found all these tools pretty easy to use. So the first one I always use is Snap Gene Viewer, because when you get a Sanger sequence back, what you get is a chromatogram. And the chromatogram is a graph that is the graphical representation of what the lasers and the sequencer saw when the DNA ran through it. And then you use a chromatogram viewer to turn what the lasers saw into actual DNA base pairs. And Snap Gene Viewer is a good free tool to do that. Once I have my sequence from SnapGene, then I open up the browser tab and go to NCBI Blast. So that's run by the National Institute of Health, the United States government. And it compares my sequence with everything else that's ever been uploaded to GenBank. And then what I do is I look at the spots that my sequence differs from the closely related sequence. And I go back to Snap Gene Viewer and I look at those spots in the chromatogram and I try to see, was this some kind of error or mistake, or is this a real difference? And so that helps me make my sequences more accurate. And so I'm not just kind of getting artifacts that shouldn't be there, but it helps me actually see what the real differences are. And so I know that when I'm seeing differences, it's not just a little bit of noise in the system, it's an actual evolutionary difference. Then once you have fixed your chromatogram and it's as accurate as you can make it, usually that doesn't take very long because usually these reads are pretty accurate. Half the time there's no errors or whatever that I can find. The other half of the time it's like maybe there's one or two, so it just takes me a minute. Sometimes the read comes back really dirty, and if I want to fight with it for half an hour, I can pull data out of it, but usually it's easier just to run the sample again. In any case, once I get my data back, then next thing you usually want to do is build a phylogenetic tree. and that shows you how everything is related. And it gives you a whole lot more information than just blast results, and a much better view of what actually happened. So it's a really good idea to not stop with blast and do go on to build a phylogenetic tree. There's a few different ways to do that. The easiest way to do that is just to upload your DNA sequences to France. And that's with this website called phylogeny.fr. And what this website does is you take a collection of DNA sequences, you upload it there, and it calculates how all of the sequences are related and does a whole bunch of these complicated mathematical algorithms, and then makes a tree for you. And I find that one-click mode works very well. So like the name implies, you just upload it, click go, and then it makes a tree. It's super easy. But you can also do it locally on your computer. To do that, I often use Mega 10, or now Mega 11 is out, to make a quick tree. And that's pretty good software, but you can't really tweak the algorithms a lot. So if you're doing a publication quality thing, then you probably want to use a more professional program. But if you're just going to post a tree to Facebook, or just for your own knowledge or something quick, just to figure out how things are related, phylogeny.fr or Mega 11 is really good. Another program that all the professionals use is called Genius. It's like $500, and so I don't use it, but you can. It's got the free 14-day trial, and it's not totally dead after 14 days. So it's a good it's a good software, but you can use free software to do everything that Genius does. So NCBI BLAST compares your sequence with everything else in GenBank, usually gives you the 100 most similar matches. And this is where you start learning about what you found. So the first thing you'll see is 100% matches, and that means that this same thing was found. And then after the 100% matches are done, you'll start seeing 99% matches. And that means that just a couple bases are different between your sequence and what's in GenBank. And this could be a closely related species, or it could be your same species, but there's a little bit of noise in the sequence or some ambiguous bases. So then I start looking at the alignments to try to figure out, is this a real difference or is it not a real difference? But then when it gets down to the 98, 97% matches, those are almost always different species. So it's much less ambiguous the further away your DNA sequence is. So here is one of these alignments that I got from BLAST. The most important number here is identities, and that means how many characters of my sequence lined up perfectly with the characters of the other sequence that it's in GenBank. And so in this particular one, 628 out of 635 characters were the same. So there were 7 differences. And so now if we look here, we can see where those 7 differences are. The first difference is this C. And so my sequence has a C, where the sequence in GenBank has an A. And so because there is that difference, it didn't put this little vertical line, the pipe character there. So I can see that's the first difference. Here's the next difference over here. I got a G where they got an A. Next difference is a gap where I had a double C, where they had a single C. These gaps are a little bit less important, a little bit less likely to indicate a real evolutionary difference. Here's one where I got a C where they got a Y. A Y is one of these ambiguity codes, and Y means that it could be either C or A. So this is not necessarily a difference. It's just indicating that they got a double peak on their chromatogram over there. And so I would check my chromatogram for double peaks, because maybe I can put a Y in my sequence too and make it more accurate. So what I do is I hit Control F in my browser and I search for the characters pipe space. And searching for pipe space highlights the differences, which makes it so you don't have to scan with your eyes through the whole sequence. Like in this one, there's only 2 differences. 776 out of 778 are the same. So searching for pipe space shows me the differences. And the first difference, I got a C where they got a K. Well, K is one of those nucleic acid ambiguity codes. And so this is not a real difference. And then here's the next difference down here. I got a T where they got a Y. Again, Y is an ambiguity code. So my sequence actually matches 100% perfectly. It's just that there was a couple ambiguous spots that should be ignored. And we can say, okay, I got the same species as these guys had when they uploaded this sequence. Here's another alignment, which is the exact same species. 571 out of 574 matches. Matched, that means there was 3 differences. But then if you're wondering like, okay, well, is this a different species? Do I have a new species or do I not have a new species? There's a couple things that immediately stand out to me that indicate that no, this is not a real difference. This is not a new species. These sequences match perfectly. One thing to keep in mind is that sequences start out very dirty and then the data gets very good and then they end very dirty. So what you should do is trim your sequence. You trim the dirt off the front and trim the dirt off the back. So the data you're uploading to GenBank is very high quality data. However, a lot of people don't do that. Maybe it's some grad student that didn't know or they were lazy. And so you can see that all 3 differences in these 2 sequences are at the very end. And that immediately makes me very suspicious because if these were real differences, there'd be some differences in the ITS1 right here. And then the 5.8S, which is very conserved right here, wouldn't have any differences. And then you would have some differences in the ITS2. But these differences are right at the end, which means it's in the LSU. So this is just, they had a bad quality sequence and it's a perfect match. So the first thing everybody always asks me is, how many differences do you need before you call something a different species? And there is no answer to that question. You can try to answer that question, but the best answer is that the more differences you see between 2 sequences, the more likely that they are different species. But sometimes you need to just give a number, like if you're just doing a massive analysis. So if you really need a number, I think 99.6% is a reasonable number. Though, sometimes, with that cutoff, you'll have some cases where you have different species that you're calling the same, and some cases that you have the same species you're calling different. But if you need a number, I like 99.6 for ITS or 99.8 for LSU. The percentage is higher for LSU because it's more conserved. But I don't just look at the DNA sequence. I also look at the morphological features, the chemical features, the habitat, the distribution. And I consider all of those factors before I decide if 2 things are different species. But when you have a DNA sequence that's consistently very different, then it's a very easy decision. I'm like, okay, this is definitely different because it's been, it's been diverging for so long that it's real easy just to know, okay, you say, yeah, that's different. If you have like just 1 or 2 differences in the sequence, then I would want to see some microscopic differences, maybe some morphological differences, some taste, smell, something else to kind of validate that it's actually different. And if I can't find anything different, I'll say, okay, these 2 species have this one species can have a variable ITS sequence. But if I do find differences and they correspond every time to these differences in the sequence, I can say, oh, okay, I can use these tiny differences in my sequence to differentiate these hard to differentiate species.
Do you have any advice for people to get started on how to learn to read these things?
Mandy asked if I have any advice for people to get started on learning how to read these things. And the way I learned is just by doing it. And I think that's a really good way just to jump in and do it. And you don't even need to sequence anything to jump in and do it, because you can just download sequences from GenBank and start analyzing them. And so you can just pick your favorite mushroom or your favorite genus, go into GenBank, search for those species, download them all, just download all the sequences and start making phylogenetic trees. And then you can kind of get some practice doing it. Or you can just make some of your own collections, do your sequences, and find out, try to figure stuff out, post it on Facebook, and maybe everyone will be like, no, I don't think this is real, or yes, great, excellent find. They usually say one of the two. But after you do that a few times, you'll start to figure it all out. It's not really that hard. Like with Psilocybe agerescens and Psilocybe suberuginosa, the ITS is only 4 bases different in ITS. So that's kind of a borderline case. You could say that maybe they're the same species. You could say that maybe they're different. And there's definitely lots of people who hold each of those opinions. And, really species are human concepts. And so the mushrooms don't care what they're called, but we like to assign names to everything. But everyone's species concept is kind of like their own species concept. Nobody can tell you what your species concept should be. You can decide that you want to see more differences before you call things different species. That would make you a lumper, or you can decide that any little tiny difference means you should call it a different name. That would make you a splitter. Both ways are correct and good ways to think about stuff. But at the end of the day, you just gotta pick something and go with it, because that way you have a name for the mushroom you've been finding, which is kind of like your goal in the end. Here in the United States, we have Psilocybe hopeii and Psilocybe mescaleroensis. These are 2 recently discovered psilocybes from the southeastern United States, Arizona and New Mexico. They have the exact same ITS sequence, the exact same LSU sequence, the exact same RPB1, RPB2, TAF1. So you can't tell them apart by sequencing, but one of them has an annulus, the other doesn't. And one of them has pleurocystidia, the other doesn't. So I can tell them apart pretty easily with the field or the microscope. So even though they have the same sequence, I would call them different species. Sometimes you'll have one species with multiple sequences, kind of like one car could have a bunch of different license plates. That happens occasionally, but it's not very common. Both of these situations are actually pretty rare. So 99% of the time, ITS sequencing will give you a really good answer. And then 1% of the time, you'll have one of these interesting situations where you have to make more judgment calls. So when you're comparing sequences, I load them into FASTA files, and that's FASTA files are a collection of DNA sequences. They're text files, so you can edit them with WordPad, Notepad, Microsoft Word, by Emacs, whatever text editor you prefer. And they start off with a right bracket, then they have a name, and then they have on the next line, you have the DNA sequence. So here's a simple FASTA file with Amanita
Can I just
muscaria.
interrupt for a second, Alan?
Yeah.
Dr. Sapphire McMullen Fisher has just provided some feedback. I don't care if you don't know if you'd like to comment. Was that different groups have different cutoffs, and her suggestion is to calibrate to genus before deciding a percentage cutoff. Arbitrary cutoffs, like suggested 99.6%, are not recommended. Multi-gene phylogenies, as well as macro and microscopic characters, are needed, as you suggested.
Yes, that's all extremely true, and I completely agree. In some genera, you'll notice that species that look very different will have very similar sequences. Whereas in other genera, like in Hygrocybe, when you have different species, you'll see a ton of differences. So it really, when you're trying to decide what cutoff you should use and whether you should call your species the same or something else, you need to look at the other species in that genus and see how far they are apart when you're kind of making this decision.
Thank you very much.
And then multi-gene phylogeny is definitely the way to go. Especially if your question is how do these species all fit together? If your question is like, how many different species do I have? Then an ITS phylogeny is pretty reasonable, because you don't really care where everything fits in. You just want to know like how many do you have? And usually if the ITS sequence is the same, then the other genes will be the same too, but that's not always the case. So it's always better to get more data. So if you have unlimited funding or a good amount of money, and lots of time, you should always sequence more genes. Though often, like in my lab, I have 5,000 bags of mushrooms, and I'm lucky to be able to get 50 ITS sequences done per week. And so if I did multiple genes on all of those, I wouldn't learn very much more, and it would take a lot more time. So I usually do ITS as a first pass. And then if the ITS shows like, oh, this thing is probably not in the genus everyone thinks it is, Then I will run all the other genes, LSU, TEF1, RPB2, RPB2, beta tubulin, and a few other ones, and that will give me a lot more confidence to be able to build a phylogenetic tree and figure out what genus something really goes in. So definitely agree very much with that comment.
Thank you very much.
Yeah. So here's a quick FASTA file. And once you have a FASTA file, the next thing to do is to align it. So alignments means that you align your sequences vertical so each base can be directly compared. You can automatically do it with muscle. You can manually do it using the space bar. So here is a little screen capture from Mega 10. And these are just a bunch of ITS sequences that I loaded into the alignment. And you can see that they're not aligned right now, which means that they're all the bases are scattered around and you can't do any kind of meaningful comparison. So if I go to the menu here and tell it to align it through muscle, all of a sudden they look like this. So that's a lot different. And all of a sudden you can compare and you can start to see what the differences are between the sequences. For example, these two sequences down here are probably in the same group, probably closely related because they share this TG insertion that all of the other sequences lack. And then these four up here, probably the same because they have this A here where everyone else has a G. So what these phylogeny programs do is they look at all of the different differences in your alignment and they calculate how something probably evolved. So the BLAST results are really cool, but you should really build a phylogenetic tree because the BLAST results can be misleading for a bunch of different reasons. And a lot of those reasons become very obvious when you build a phylogenetic tree. The most common way people misidentify mushrooms using DNA sequencing is to blindly trust the names that are in GenBank. So anybody can upload a DNA sequence to GenBank and they can attach any name to it. And that means that some of the DNA sequences in GenBank are very carefully named, they're accurately studied, they used a microscope, maybe an expert in the field uploaded them, and you can really trust those names. There's also a lot of type sequences. So those are when the holotype gets sequenced, that's the gold standard. You know those are named right because it's the holotype. However, a lot of people have like grad students, and these grad students are just kind of like uploading on, you tell them to upload a whole bunch of DNA to GenBank, but what they really want to do is go out and drink beer and party. So they upload them real quick, but don't really verify the names. And so stuff gets uploaded to GenBank without having proper names. Or with having wrong names. Also, a lot of times what somebody will do is they'll be like, I want to sequence everything in my herbarium and upload it to GenBank. That's a really awesome thing to do and it's very noble. However, many things in the herbarium are mislabeled, they're wrong because the collector maybe wasn't very skilled. It was just, some kid just, collecting mushrooms, deposit in the herbarium, and then it gets into GenBank with the wrong name. And then sometimes you have just like the tubes got switched, so a pipetting error, or when you're copy pasting, you thought you copied, when you pasted, you have a data error. So sometimes your sequences are switched. Anyway, the point is you cannot trust the sequences in GenBank to be accurately identified. So if you do BLAST and you see you have a 100% match and the 100% match has a name on it, that only means that what you found is the same as what they sequenced. It doesn't mean that you're fine. It doesn't mean this name that you got is correct. So to validate that that name is correct, you should look for more matches in BLAST, which is good, but it's even better to download all those matches and build a phylogenetic tree. And the really valuable thing that shows you is that like say you get 100% BLAST match to Rusula brevipes, and you're like, awesome, I found Rusula brevipes. Turns out, no, you probably didn't find Rusula brevipes because there's about a dozen species going under the name Rusula brevipes, and the real Rusula brevipes has a very small distribution. It was described from New York, United States, and it doesn't occur in most of the United States or most of the world. But if you find a white Rusula and you sequence it, very likely your closest BLAST match will be Rusula brevipes, and that'll be wrong. But what you should do is download all these 100 BLAST matches, calculate a phylogenetic tree, and when you do that and you look at the tree, you will notice that Russula brevipees doesn't just show up once, it shows up in 7 or 8 different places. And then you're like, oh, okay, that puts this data into context a whole lot better. And I shouldn't trust this name because this name is being applied to a whole lot of different species. And that's super valuable information. So when you calculate a phylogenetic tree, sometimes you calculate bootstrap support, and that tells you how confident the computer is that it has put each branch in the correct spot. So higher numbers are better. And if your numbers are low, that means that your sequencers are not aligning, and your tree is still valuable, but the computer is just letting you know that it probably is putting the branches in the wrong spot, and they didn't really evolve like that. That's really valuable information. And the best way to fix that is to sequence more genes. And so if you sequence some more conserved genes and concatenate those sequences with your ITS sequence, you get the best of both worlds, because you have the species level resolution of ITS with the more conserved genes that help you get better bootstrap support. Especially if you have diverse species in your tree, then you'll have a really nice tree that's both accurate at the species level and at where it's showing you where everything gets placed. So here's a little tree I made. This was a little tiny bright red Cortinarius that I found in Mexico, and I sequenced it, and I see that it's its sister to this other species over here. And one thing you notice over here is that there are two things on this branch, and they have identical sequences, but they have different names. And so you know that one of these names is wrong because they're on the same spot, so they can't both be right. But one thing that I notice is that Cortinarius rubobruneus is a type sequence. So this is a holotype. And that means that this bottom name is correct and this top name is wrong. That makes sense because Dermosobe phoenicia is a name that got applied to a whole lot of different species. And one of those species that the name Dermosobe phoenicia got applied to is Cortinarius rubobruneus. So that's a cool thing that I learned by making this tree, and that my species that I found in Jalisco was sister to whatever this is. And then down here, a little bit more distantly related, you have all these other species. And a really cool thing to do when you're building trees is to put locations on them. Your trees all of a sudden get a whole lot more meaningful when you do put locations on the trees, because all of a sudden you can see like, oh, a certain clade is in this part of the world, another clade is in a different part of the world. And that's a really cool thing to be able to see in a tree. It's a little bit time consuming because you have to look up the location of each sequence, but it's definitely worth it. So that is all the slides I have for you. Here's a couple of resources. We have this fungal sequencing Facebook group. We have fungal sequencing results where you can post your sequences that you found. Phylogeny.fr is really cool. Even newer is ng-phylogeny.fr, which gives you a little bit more control and uses some newer algorithms. I use them both. I actually like the old one a little bit better than the new one. But give them both a try, see what you like. And then you have this GeneWiz is one of the many Sanger sequencing places in Australia. Probably want to use Macrogen in South Korea. And then if you're wondering how all of this stuff works, you can look at the polymerase chain reaction Wikipedia article, and it kind of lets you know how all this DNA copying works. So hopefully you have some questions about all this, because I know I covered a lot of stuff. And just going from 0 to 60 on this is going to take you more than an hour. So I am happy to take questions now.
Wonderful.
So perhaps you could unmute everybody.
Certainly.
Or maybe we should go to the chat. Maybe people. Sorry, go ahead. So first I will flip through the chat and answer the questions in the chat. So the Facebook group for fungal sequencing is facebook.com groups.
So I have
Sorry, go ahead.
just a question. So when you're how long does it take, I guess, to do from beginning to end? You've got your mushroom specimen and you're starting this process. What sort of time investment does it take from beginning to end?
Well, if you have just one mushroom that you want to sequence, it is 2 minutes of work, 10 minutes of waiting, 2 more minutes of work, 2 hours of waiting, 5 more minutes of work, 20 minutes of waiting, and then you mail it off. So kind of like one afternoon. But it's way more efficient if you do more at a time, more samples at a time. So I usually do batches of 16 or 24, and that's maybe about 3 hours of work to get those 24 sequences. If you wanted to do 96 sequences, it'd probably be about 6 hours of work. And then once you get your PCR product, you drop it in the mail, or a lot of these places, if you're in a big city, they'll come pick it up, send a courier and come pick it up. And then once it gets to the lab, then they run it and your sequence comes back the next day. So like if I start in the morning and I'm able to get it over to the lab, I'll have my sequence in about 18 hours. But usually I just kind of like wait a couple days. But so usually I get my data back in a couple days. Or if you have a whole lot, maybe they pile up and it takes longer. But that's about how long you can expect.
Fantastic. Wow, this has been a really huge knowledge transfer, and you must be exhausted after sharing all of that information and for talking so long. Thank you so much.
No, not really. Actually, I get energized by telling people about this stuff because I really want to excite people and tell people about it. And it's not every day that I get a chance to talk about this nerdy stuff to people who actually are interested in it. So I'm super happy to answer questions either now or later. I'm easy to get a hold of.
Wonderful. Well, we may not hold you too much longer. I've got there's an outpouring of thanks, of course, in the chat comments. Everyone's really enjoyed the presentation. And again, thank you for making yourself available to contact if anyone has any further questions. Of course, you can relay questions to us at info@queenslandfungi.org.au, and we'll share them with Alan, and we'll get back to you. Or there you go. Alan's shared his email as well too, so you may contact Alan directly as well, everyone. Thank you so much, Alan.
Facebook message was great.
Excellent.
Thank you so much.
Oh, hang on. We have a question from Eric.
Okay, awesome.
Any different DNA yield results on different parts of the mushroom, stipe versus cap?
No, the whole mushroom has the same DNA in it. So you can use any part of the mushroom. One strategy works well with thicker mushrooms is to break the mushroom open and taking something from the inside of the cap or the gills or the stem, because that way you know that there's no contaminants from other mushrooms like rubbing against it. But honestly, that's not much of an issue, and so I don't usually do that. Usually I try to grab DNA, grab a little bit of the gills, because there's a rumor that the gills have a little bit more DNA because that's where all the reproduction is happening. But honestly, it doesn't really make any difference. If your mushroom started to get a little bit moldy, then whatever part of the mushroom dried out quick, usually the very edge of the cap, if that dried out first and then the rest of the mushroom kind of got a little bit rotten before it could dry out, then the edge of the cap is going to be the best part.
Very good. Thank you very much. Vanessa, did you have a question? I couldn't see your question in the comments. Did you want to unmute and ask your question? No? Okay. Tammy, unmute is blocked. I beg your pardon. Okay, so Fran had a question as well too regarding repeat extractions and whether altering reagents will improve success. I think she reiterated in her question that, where is Vanessa? Oh, sorry.
Yeah, so the easiest and the least work thing to do is just going to be to take the same DNA extraction and run it again. But maybe the same problem happens the next time, maybe it doesn't. So often if a PCR doesn't work, I'll try a few different things. One of the best things to do is to dilute your template 10 times into 100 times. And if the problem is PCR inhibitors, that usually fixes that. So I'll just run a few different PCRs with the same sample and try it a bunch of different ways, and whatever gives me the strongest band is what I send off for Sanger sequencing. Another thing that's very likely to work is to do another DNA extraction. So just grab a different part of a mushroom, or if you have a collection with multiple mushrooms, just grab a different mushroom of the collection, run your DNA extraction again. That's very likely to work. Another thing that is more likely to make your PCR work is lowering the annealing temperature. So the annealing temperature determines how tightly the primers stick, and it determines how well the primers need to match the sequence in the mushroom in order to bind and stop the copying there. So the lower your DNA annealing temperature is, the better chance that the primer will stick. So usually if you do 55, if you bump it down to 52 or 53, then that makes the primer stick tighter, and sometimes that will make it work. And then sometimes, no matter what you do, it just doesn't work. And that could be because maybe your mushroom got a lot of bacteria or mold and it ate all of the mushroom DNA, or maybe it got heated really hot, like way hotter than boiling, and that caused the DNA to all break. Maybe there's like a PCR inhibitor. Some samples are just very PCR resistant, and sometimes I figure out why and sometimes not. But fortunately, that's like 1% of the samples that I just can't make work. And then the rest of them, I'm able to fiddle with some parameters and get a good sequence out of it.
Right, thank you. Fran, did that answer your question?
Yes, it did. Thank you very much.
Okay, no worries. Vanessa, you had a question for Alan as well?
Yes. I collect fungi for the herbarium, as most of us do here. I'll probably never actually do DNA stuff myself, but I would like my specimens to be in the best situation, so if someone in the future wants to do it. What would be the best way we can do that?
Well, there are a few different ideas about the best way to do that. Usually, most specimens work. I've left specimens on the dashboard board of my car for a year and then sequenced them. And even though they were baking in the sun for a year, they still work just fine. But then sometimes specimens don't work, and you don't know why. You know, PCR is not going to explain to you why it didn't work. So people kind of get a lot of superstitions about different things that they should do. One of the most common ones is drying temperature. You know, anytime you have really high temperatures, you're going to break down the things more rapidly. So general wisdom is to dry your specimens at relatively low temperatures. So I usually set my dehydrator for about 40 degrees Celsius. So that doesn't really bake them. And so that's going to increase the chances. Even more important than drying temperature is to keep the specimens extremely dry. So they should be cracker dry before you take them out of the dehydrator. It's a really good idea to throw a little desiccant pack in there. And also just make sure the humidity in the herbarium is carefully controlled so they stay dry and nothing can grow on those mushrooms. One more thing you can do is keep the mushrooms at low temperature, and that's not really practical for a whole herbarium, but if you know you have something that someone's going to want to sequence later, what you can do is you can dry the mushroom, or even before you dry the mushroom, when it's fresh, you can break off a little tiny piece, throw it in a tube, and put that in a freezer, maybe even a -80 freezer. If you use very small tubes like the PCR tubes, the 0.2 milliliter tubes, you know, you can have hundreds of tubes taking up very little space. And when you lower the temperature that much, you're slowing down all the enzymes and all the different chemical reactions. So if you keep something in a -80 freezer, I don't think you'll have any trouble sequencing it 100 years later. So that's another strategy, is to save a tiny bit of the collection, either in the freezer, or you can use a stabilizing solution. Some of these buffers, like EDTA, is commonly used. EDTA is a chelating agent, so it chelates the metals. Turns out these enzymes that rip the DNA apart, they need metals to be able to work. So if you chelate all of the metals, then that keeps the DNA stable for a very long time. So you can buy like commercial DNA stabilizing solutions, and you throw a little bit into one of these solutions, and they do their best to basically stop time. And that can keep things in good shape. for DNA sequencing down the line as well. Though I have, tried sequencing pretty old collections, and sometimes they do work. So you'd be surprised. But anything you can do to, lower the temperature, keep them dry, and not let them get moldy or bacterial before they do dry. So don't let them sit around at room temperature, growing bacteria for a couple days. Anything you can do will be very much appreciated by the people doing the DNA work later on.
Thank you. It's great.
And Vanessa, I absolutely agree with everything Alan said. This is Sapphire. If what I recommend people do is, I think the whole tube, I don't think people have a -80, but I'm happy to send people some tubes and some silica. And you make sure they're silica dried, and then, because particularly in Southeast Queensland, but also tropical Queensland, humidity is a nightmare. And I've lost more collections than I'd like to think about, even with silica and various things. So taking a couple of good clean samples, have a couple of tubes clearly labeled, use a bit of color change silica so that you know that sample in there. And then it's so much easier the other end. And you can do all of your DNA taking quickly. Take an hour and you've got 5 samples or 10 samples. You take them all before you mess around with them. They don't sit on the bench for, until after dinner time when you get to look at your microscope. If you take your DNA, I've now done lab shots, and DNA are my first priority. And if I don't get to do lab shots, DNA is just the first priority.
Thanks
That
for that,
is
Sapphire.
really-
That's excellent. I think, Sapphire, too, I think the challenge with humidity, you may have mentioned to me a little while ago, drying specimens at 40 degrees or lower in a really humid environment may also require coupling a dehydrator with a dehumidifier in the room at the same time.
I had a dehumidifier under my dryer when I lived up near Noosa.
So you're looking at, a dehydrator and a dehumidifier to successfully dry specimens in a really humid environment.
Yeah, that is really good advice. Especially if you have a tube of silica gel, the color changing silica gel, so when it needs to be baked again to bake the water out of it. It's just saving a tube, because it's a super good idea, because once the mushroom gets moldy and bacteria, then getting a good DNA sequence becomes much more difficult. And I have successfully gotten good DNA sequences out of very moldy mushrooms because I use primers that ignore ascomycetes. But once the mold grows too much, it's just going to metabolize all the mushroom DNA, and you won't be able to get sequences. So storing a tube, even at room temperature with some silica gel, so you're just keeping a few milligrams very dry, that's a really good idea. And then if you can throw that even just in a regular home freezer, -20, that will slow down the reaction many times. And so, -80 is of course best, but in practical terms, -20 is almost probably 99% as good as -80 for keeping these dry specimens in really good shape. So when you do the sequencing later, it works.
Okay, since we're talking about best techniques for drying, Warwick has a question regarding freeze drying. Have you ever had any experience or feedback regarding freeze drying and its success or failure?
I've never tried it, but I think it should work extremely well. My lab just got some freeze dryers, and I'm working on fixing them up, and so I'll be able to try that. But I think it's a really good idea. And a lot of times you'll have like a very tough mushroom, like a polypore or a lichen, and the freeze drying makes them very brittle, so you can powder them. And so that is really cool. But also just being able to get rid of all the water without needing high temperatures, I think that would be a really good storage method. So I think if you have a freeze dryer, you should give it a try. And I would say,
if I was to break that down for a layman and said, how would I freeze dry at home, or someone who is just a simple person wanting to freeze dry? Initially, someone said to me, all you have to do is leave your mushrooms on a rack in the freezer and they will just naturally freeze dry. Is that a workable or a viable methodology for freeze drying?
And also just be careful. Some microscopic characters get lost if you freeze dry. So it might be good to do the DNA freeze drying, but you don't want to do your whole herbarium collection freeze dried. There are I've looked at some awful freeze dried things, and spores were broken and all sorts. So yeah, don't
Right,
do
okay.
it
Yeah,
for the whole
that's-
Very
sample.
I was
good.
about-
Vanessa's also saying, wouldn't freeze drying destroy the cell structures?
Mhm.
So I
And that's
think we're on the
what I
same
was
page.
about to say too, because whenever you freeze something, these ice crystals are like little blades that just slice everything. And so the DNA will be kept in good shape in the freezer, but the microscopic features in your collection will basically be ruined if you freeze it before you dry it. So yeah, definitely just freeze dry a little bit. And sometimes I'll even just freeze fresh a little bit. Just take a little bit of fresh mushroom, throw it in a tube, close it up,
Yeah.
put it in the
That's
freezer,
great.
and
Works really
that
well.
works well too.
Yeah.
Okay, so not a comprehensive process for preservation, but a selective process for preserving DNA is to freeze dry some of your material. Warwick's also suggested that snap drying would preserve the cell structure. Snap freezing. Sorry, snap freezing.
I don't know what that is.
Yeah, no, me neither.
Doesn't it involve liquid nitrogen?
I think snap freezing is immersion into liquid oxygen or something like that, isn't it?
Nitrogen.
Well, yeah, liquid nitrogen is the thing that has decimated the microscopic structure. So I think liquid oxygen would be similar in decimation.
Okay, no worries. So good advice there. So and Vanessa agrees. So a small part of the specimen and really with a view to preserving DNA, but keep being mindful that cellular structure or microscopic features could be lost for that specimen if all of the specimen was processed that way. So probably drying, probably freeze drying, and also fresh frozen with some sort of primer as well too would be a comprehensive collection process.
I'm not really sure that fresh frozen is the best idea. I mean, it's worked for me, but I don't know. I think it'd be a good idea to do as many different ways as you can, and then just see how many works. Really, just if you're processing your samples within a year, just drying them is fine. Though, like you said, if you have a really humid environment, a lot can happen in a year. And a lot of times you'll have very cracker dry mushrooms and you put it in a Ziploc bag and seal it. And if you take that in a humid environment, the moisture will have no trouble going through that bag. And when you go to use your mushrooms, they'll be moldy. So that's the main thing you got to worry about. I live in California.
Silica gel
where
would
the
come
air
in
is
very handy with good quality and a good amount of silica gel.
Silica
with the
gel
dry
is
specimen.
really important. Yeah.
What I noticed and what I reminded myself is very important is to put the silica gel in the dryer with the specimen, because you don't know if it's expired or not unless it's a reactive type of silica gel. So put the silica gel with the specimen in the dryer and bag them up both together out of the dryer, so you know the silica gel is dry and ready to absorb any residual moisture.
That's really good advice. And also, I just check on my specimens every once in a while and make sure that silica gel is not changing color in the indicator strip. And if it does, then throw it back in the dehydrator.
Yes, I've just reprocessed some specimens after 6 months and they weren't as crisp, and the silica gel had turned. So the specimens in the bag, silica went back in the dryer and then got rebagged up. So it was a good follow-up thing to do. We do have, additional questions. Cloning would preserve DNA, only need a small amount of tissue. That's a suggestion from Wendy Ring. And we certainly had some feedback from people who are, I guess, getting clean spores and putting them onto agar plates or whatever they are. And they get a mycelial growth, and then that gets frozen. So that's not a bad methodology
That's
too.
a really good idea. I find that I get very good sequence results when I sequence a fresh culture. So if you can clone the mushroom and keep that culture, maybe in a culture slant in the fridge or something, keep it going, then that's a really good way to keep, because then your DNA is always fresh because it's alive. One thing to keep in mind is that agar is a PCR inhibitor. So when I scrape up a little bit of culture from an agar plate, I try not to scrape up any of the agar. Or instead of using agar, if I use gelling gum, which also gels up plates, that's not a PCR inhibitor. So I sometimes use gelling gum instead of agar if I plan to sequence stuff, though I've really had good luck sequencing from agar plates too, just by being real careful about not scraping up too much of the agar when I'm doing it. But also, spores can be viable for quite a while. Some people say even like 20 years at room temperature, although usually when I try to use spores that are a few years old, they don't really germinate. But probably, spores definitely last you a year or two, and probably longer in the fridge. So, saving a spore print is a really good idea as well, as so you have a backup in case your culture dies or gets contaminated.
Yes, very good. And of course, a spore print is a very important part of the collection. Wendy Ring also mentioned liquid cultures from spores.
I think it might be best to go to agar first, so you have a clean culture, and then make a liquid culture from your agar. But yeah, liquid cultures can stay around for a real long time, and so that's a good way to do it. Also, when I culture stuff, I almost always sequence what I culture, because very often, especially you're cloning a fresh mushroom, what grows on agar is not your mushroom, but something else in the environment. And some contaminants are very obvious that make these crazy forms on agar. But if you have just like some other mushroom, they kind of all look like white tomentose growth on agar. So I will scrape up a little mycelium and sequence it, so I know that what's growing on my agar plate is actually what I cloned. And I would say 70–80% of the time it is what I thought it was, but a pretty good percentage of the time I'm very surprised with what sequence comes back. And if you were to take that culture and try to grow it, you would probably colonize your grain and never fruit, and you would be like, man, I got a non-fruiting strain. And that wouldn't even be true. It's just you're growing something that's, some sort of soil fungus.
Great feedback. Thank you. Culturing and cultivating, of course, is a whole world that provides its own fresh set of challenges. So launching yourself into that space could provide you a fresh hell. But if you like a challenge, I guess you would go there. It's also great information too about the agar and the potential impact it has on DNA sequencing. So thank you for that.
Yeah. And also, the sequencing for people that like to grow mushrooms, if it only costs $10 to sequence, if someone's growing a bunch of mushrooms, $10 to know what it is that they are growing is really cheap, really cool information that's not too expensive.
Excellent. Does anyone else have any other questions? I think we're all good. Look, thank you so much, Alan, for opening your mind and sharing your knowledge. It's a wonderful thing for fungi and a wonderful thing for conservation that someone as well-resourced in terms of his understanding of this process is prepared to share so much information with us. So thank you so much for coming today and for allowing us to capture that content. It's been just enormously enjoyed by everyone, and especially by us who are more deeply immersed into mycology and specimen collecting. So this has really been valuable to us, and I thank you from the bottom of my heart. Thank you so much.
Thank you. You guys have been a great audience. So definitely contact me with any questions, and hopefully some of you guys can get some of this PCR working at home, and I'm totally happy to help you get it working reliably.
Awesome. Well, if there are no more gold nuggets about to fall out of your mind that we need to capture, I'll stop recording.
I think we got most of it.
Okay.